like that. Okay, I start immediately. Thank you very much for inviting me for this webinar. It's the first time I present at a webinar, so we'll see how that goes. It's very different, I have to say. I will give you a short background. I start with, I call this presentation the first step towards a vaccine, because there's many steps that has to be passed, and we take the first steps here. I will start with a background a little bit about Red Glead Discovery. We are a contract research organization that was founded in 2011 by 7 people who used to work at AstraZeneca in Lund, but when AstraZeneca decided to close the site, these people decided to stay and start their own CRO. In 2013, the first employee was hired. 2014, we acquired another company called Peptide that was producing peptides or peptide research, and that gave peptide chemistry capabilities to Red Glead. 2015 was the first spin-out collaborative project. It was a new company started from research from the company in Red Glead and some university. And then in 2016, we licensed a fragment-based lead discovery platform together with another CRO called Ceramics Biostructures. And last year, we acquired the company VAC, which is the fragment-based lead discovery platform. So that are some highlights in our company. We also is one of the fastest growing Swedish life science companies. So what we do is that we have a drug discovery service that we focus on small molecules. Sorry?
Would you mind to turn on your camera so we can see you?
Absolutely. Let's see where I can find that. There. Hi, everyone. I'll see if I can.
Please go on.
Adjust that a little bit. Okay, thank you. So, drug discovery service focus on small molecules and peptides. We have a milestone and data-driven project progression, hypothesis-driven medicinal chemistry, and integrated drug discovery offers. And to the left here in the screen, you can see a little bit of the 5 major parts that we offer. We have medicinal chemistry and the custom synthesis. We have peptide chemistry. We have in vitro biology and ADME analysis. And more than 200 clients we have served worldwide since we started in 2011. And this is us. More than 30 people working now. Today, we are 33. On Monday, we are 36. So we are still growing. A lot of experience from different pharma companies, a lot of expertise in different target classes, therapy areas, administration routes. Since we have a lot of different parts in the company, we can do a very integrated approach between medchem and ADME, PK, biology, etc. But that about this is more. Sorry. This is more how it looks in the early drug discovery where we're working. So from early to late drug discovery, farm dev support, we do everything preclinical and nicely color-coded with our different teams. But today we will focus just on the peptide chemistry and the peptide drug discovery, because that's where the interest is at the moment here. So we start to talk about the research towards a vaccine. And before I go further into that, I need to thank some people. And I need to thank Birger Sørensen and Andres Susrud from Immunur, and Samara Mamidi from Red Glead Discovery. Samara has been doing almost, in principle, all hands-on work, and the Immuno company here, I will explain a little bit more why they are here. So Immuno is a Norwegian-based biotech company. They've been around since the 1980s or something like that, and they've been focusing on vaccine development since 20 years back. They currently have 6 projects ongoing in different vaccines, and we have worked with them on and off since 2012 in different projects with them. They have a platform technology they use to identify and design peptide domains to use for vaccines. And then it's going to be a peptide-based synthetic vaccine. The type of vaccines that have been, especially the synthetic vaccines, have been more and more interesting, especially since the swine flu outbreak in 2008. So Immuno is our client in this project, and we are helping them along the way. So if we start talking about the bad guy in this whole thing, is the SARS-CoV-2 virus. Looks nice like this. Nicely a picture from Wikipedia. And what happens is that we have the spike glycoproteins that bind to the ACE2 receptor. And we started to look at these glycoproteins, and of course we find some ACE2 receptor binding domains. There is a 76% homology between these glycoproteins compared to the SARS-CoV virus that was responsible for the SARS outbreak in 2002. There is a lot of HIV similar sequences in them. There is a couple of influenza A sequences also. And the more problematic part there is that there is a huge number of human-like domains in these proteins. And those could cause a problem later on. We cannot really vaccinate for human-like domains because that would lead to an autoimmune disease. So, starting with the design and the synthesis, we started with the sequence starting points for the vaccines using the platform technology from Immuno. We looked at what we had there, and we started to do some simple optimizations for stability before we did the synthesis. Some things you can do easily without having any ID or shown things. Since we think, since we have high beliefs in our project, of course, we could also start optimizing for the downstream processes. And we don't want to end up with a candidate that is going to be problematic to synthesize in large scale or purify or something like that. With this in place, we started our solid phase peptide synthesis and started producing some peptides. And we have so far produced 15 peptides. They're all between 20 and 40 amino acids. And as you might understand, I cannot go into detail of what they look like. But we have finished the first round of synthesis, and now we're going for the next step, and that is the evaluation. So we've done some physical chemistry, stability and solubility of these peptides. We are doing acute tox at the moment. So we're dosing animals to see if there is any acute toxicity within 1 week. And we have started in vitro immunization of these peptides. The next step for us now is to do a primary in vivo immunization. Currently designing that study. So that's going to be in some animal, mice or rats or rabbits, depending on, we're still undecided on that. That's also going to be done together with another CRO company. We have also initiated the GMP production, so that we will, as soon as we have our candidate, we should also be ready to go for human studies. After these tests, we will go into adjuvant testing and selection. That's a big area too, to find the right adjuvant to give a little bit trigger to the immune response that something wrong is happening. There's a lot of things done in this area, maybe not so much done systematically, but we will see what we can find. There's a lot of knowledge in this area. And when that part is done, we will go for deciding which is the optimal dose and which is the optimal route of administration. And then, of course, we have a very bold plan, and that is that we will have a first in man in August, September. That's our plan. So I'm sure there's some things I can talk about and some things I can't talk about now, but I thank you all for listening, and I'm ready to answer any questions. Thank you.
Thank you very much, Richard. We all hope that, of course, you succeed and get the vaccine ready for being used in humans. There's a question by Dionysius Sanchez. He's asking, how does your vaccine strategy compare to others around the world? So why did you decide this approach and not any?
I can't tell for all of them, but there is many different strategies in this. Traditionally, you use an inactivated virus particle. Mm-hmm. I think that could be a little bit problematic in this case. There is a lot of human-like epitopes in the proteins, so and those we will not get the antibodies for. That could be a problem. There is the Moderna is doing a mRNA-based vaccine. There is I think it was University of Melbourne that is working on using the BCG vaccine that we use for tuberculosis. I've seen some good things there, so it's a completely different route. I don't know what all other peoples are doing, but there is a lot of different approach. I think I read the other week there was 54 or 55 different vaccine programs ongoing against this. So some are probably very similar, but we're all over the range, and hopefully some of them go all the way. I really hope so.
Yeah, I mean, we all hope so that someone will find the vaccine quickly. There's another question. So you mentioned that you are using the sequence of the virus, and I know that there are different groups that have sequenced the genome of the virus. So which sequence are you using? Because there are some differences from country to country. So the virus mutates not much, but a bit. So what approach are you taking here?
We're looking at all available data, I would say. So we see, we look at all the available data, see what sequences, we see how much mutations there are in selected parts of the spike glycoprotein, and there is mutations that may or may not affect it, but we have to see there. I mean, viruses mutate all the time. Hopefully they don't mutate. Hopefully they mutate so they don't get infectious. Yeah. but we look at it and we try to follow up as much as possible to see what we can do. But hopefully we have selected good sequences with there is some mutations we've seen that have been outside what we are looking at. that there might be someone into. But hopefully there can be an immune response for the whole vaccine that we're doing that will work for several different mutated strains.
We have actually a lot of questions. Next one is from Alberta Brown, asking, have other projects within Red Glead Discovery just been put on hold, or are you having any activity outside this project about COVID-19?
We have a lot of activity outside the COVID-19, actually. As I mentioned in the beginning, we are 5 areas that working, and this is very related to the peptide chemistry. In the peptide chemistry, we have partly put some projects on hold, but the project we have put on hold is actually another project with Immuno. So all other clients could be safe that their projects are ongoing, but Immuno changed when this pandemic hit. Immuno changed the research. We had an ongoing project with them in another vaccine, but they said that we should halt that for now and focus on this COVID vaccine.
All right, I will ask you the last question. There are quite a few, so I'm sorry for those of you who won't get the answers, the questions answered. So Lasse Meyers is asking you if you can elaborate a little bit more on the targets of the peptides, or the targets for the peptide, without revealing anything. Do you have a target focus, or do you have several targets in mind?
No, there's not. I mean, the target of the peptide is to induce an immune response and produce antibodies in the body. So, in principle, the peptide's target is our immune system. So they will be the fake virus in our body that we develop our antibodies for, so that if we get infected in the future, we have our antibodies ready. We will not. But that's the target.
Okay.
I hope that answered the question.
Yeah, I'm sure it did. Thank you very much, Richard.
Thank you.
This, I will give you a big hand.
That's good.
Yeah, thank you very much for joining. Yeah, it was really interesting to hear about your strategy.